So welcome once again for the second day of ATAL FDP on drug discovery and design in 21st century. Professor Jayaram has already joined us. So I will give a brief introduction about Professor Jayaram. Professor Jayaram is a faculty at the chemistry department of IIT Delhi. And prior to this, he obtained his PhD in chemistry from the City University of New York in 1986 under the guidance of Professor David Beveridge, a renowned quantum chemist and one of the world's leading experts in DNA modeling. Dr. Jayaram's thesis work was concerned with developing methodologies to model nucleic acid constituents at atomic level under aqueous conditions on what was then one of the largest computer installations of IBM in Midtown Manhattan. After his PhD work, he took up a postdoctoral assignment with Professor Barry Honey, a pioneer in the field of biomolecular electrostatics and bioinformatics at Columbia University, USA. Dr. Jayaram's contributions on electrostatics of DNA have eventually found their way in Delphi software of what was formerly BioSim and now Accelerate. Subsequently, he worked as a senior research associate with Professor Beveridge at Wesleyan University, where he developed methodology to help understand the energetics of biomolecular recognition. At IIT Delhi, he started building the infrastructure to carry out biomolecular modeling and creating the necessary science and software, pursuing the dream of developing in silico solutions for personalized medicine, like individual specific drugs with no side effects. A result of his efforts is the supercomputing facility for bioinformatics and computational biology at IIT Delhi, and the Gene2Drug suite of software which are publicly available. So with this, I welcome Professor Jayaram for his talk.
Good morning, Professor Kumar. And good morning, everybody. So I hope all of you can hear me. And
Yes, sir.
okay, okay. Oh, that's wonderful. Well, thank you very much, Professor Kumar, for inviting me. I'm very happy to see so many youngsters participating in this workshop. It's an excellent opportunity for me to share with them some ideas coming from our laboratory and probably excite them to do more research in this area. So someday we'll see a planet that is disease-free, right? That's the expectation. So let me share my screen, and I need your help here. So are you able to see this screen here?
Yes, sir. Yeah.
Okay. So I'll go to
full screen mode, so you can go.
Yeah, I'll go to the full screen mode. Is this okay?
Yes, sir.
All right. So let me turn off my picture here so you can see. All right. So Professor Kumar, should we start? Hello?
Yeah, we can start, sir.
All right. Thank you again for the opportunity. So I'm going to focus on integration of chemistry with biology and IT towards a disease-free planet, because my affiliation is towards chemistry as well as School of Biological Sciences, and I keep calling it this as SC of Bio, that is Supercomputing Facility for Bioinformatics and Computational Biology at IIT Delhi, right? So what is a big challenge? Well, today's challenge, of course, is the big data. And we are now seeing, especially the last 2-3 years, so lots of courses and online certification programs on how to do data analytics and all this stuff. But from our point of view today, from the drug discovery point of view, what is big data in store for us? Well, we would somehow like to utilize this data to extract some information and knowledge out of it and eventually create products useful to society. That's our plan. But how do we do it? Well, it's nice if artificial intelligence programs can do all this stuff and then eventually give the products. But somewhere along the line, to create new science, you need a hypothesis, you need an idea. How do I analyze this data? What idea is buried, or what hypothesis, or what reality, or what principle is buried in this data? That sort of extraction of a principle requires hypothesis generation. This is where you cannot throw the entire blame on the computer. You have to tax your brain a little bit, right? Make sure that you do a good job of the data analysis. Stare at the data, suggest a hypothesis. Maybe 99 hypotheses don't work, but one of them works, and that will be like a boon for the society that you are really creating, and you're making a name for yourself in the textbooks. So keep this in mind. And luckily for all of you and me, most of the data is freely accessible. The good news is like in NCBI, National Center for Biotechnology Information USA, it has huge amount of genomic information, including the coronavirus information, genomic information. So you can stare at it, see what does it look like, right? How does this genome organize itself? In fact, this is one of the outstanding questions. How do you stitch together DNA or RNA to make a genome that is functional, that can do good things and sometimes bad things like this corona, right? And if you understand it, then you can tamper with it. You can tinker, you can make sure that it is safer to humans and plants, animals, kingdom, and so on, right? And then there is another database, UniProt, that has protein sequences. And I keep coming back to proteins because most of the drug targets happen to be proteins. So we need to know much more about the protein sequence as well as structure. So structural information is also available at the Protein Data Bank. We call it as RCSB, right? Structural data bank. It also has huge amount of structural information or three-dimensional structures of the protein, whereas sequences, of course, are in millions. Structures are in hundreds of thousands, so there is a big gap. In addition to this, what's more interesting, especially in the context of today's pandemic, is the availability of millions of purchasable compounds from various databases, including the Zinc database. 160 million compounds are. So in fact, some other company the other day, they were telling me that they can give us a choice of half a billion. There's 500 million purchasable compound. That's interesting. Now, with so many millions of compounds, can't even one compound be utilized as a drug molecule against this COVID? That will be a question that you will ask, a question that I will also ask. And we'll let us see how we can utilize these various compounds that are organic molecules, small molecules that are available, to utilize them to harness that power of the numbers here to be able to come up with at least one or two molecules which are ready to cure a particular disease, right? So this big data in that context is extremely useful, and it's free. So my request to you is take a look at this data, visit these databases, and see if we can generate some hypotheses and eventually create some products useful to society. And I will show you my pathway. I'm sure you are very innovative and you can come up with your own modifications to the pathway that I'm suggesting and come up with something really extremely useful to get rid of disease from the planet. Now, there are other issues like the big data, of course, will be even bigger and bigger if you added genome plus proteome plus small molecules. And if you added gene expression data and protein-protein interaction networks, we're talking of exabytes. I'm sure you understand what is this, right? Giga is 10 to the 9. Right? Tera is 10 to the 12. Peta is 10 to the 15. Exa is 10 to the 18 bytes. That's a huge amount of data, right? So let's use this big data, make some drugs, and get rid of disease. That's the goal for today. How do we do it? Well, this is my dream plan. My dream plan is that 10 a.m. the disease is reported on a smartphone these days. People can upload the symptoms and a genome card. If it's already just like Aadhaar card, very soon all of us will have probably a genome card, right? And what do I want? Well, I want that by 5 p.m. the drug is delivered at the doorstep like this, Zomato or Amazon, or somebody will just come and deliver it. That will be wonderful, right? It's 10 a.m. to 5 p.m. no matter what the disease is. And what's the plan that I am sketching here? The pathway that I have in mind is that from the genome, you identify a gene that codes for a protein, a rogue protein, bad one, and you convert this gene using Hargobian Corona's uh, genetic code to a protein sequence, methane. So here, this is ATG, adenine, thymine, guanine, cytosine. That's a DNA space. So from DNA space, you're going to the protein space, methionine, alanine, leucine, tryptophan, methionine, etc. And then you build a 3D structure. So this is a three-dimensional structure of the protein, and then you find active sites, etc., or a binding pocket or some functional site which is necessary for the function of the protein. Let us say this is a protein and you want to inhibit it, viral protein that you don't want to function. You don't want it to function for this, so that the organism just dies or goes away, gets clear from the system. So you want to find an inhibitor molecule that is a small molecule. So you have, let us say, access to these millions of compounds. So you can choose which compound will bind here and inhibit this protein. And you have this help of these various softwares and supercomputers to do this job. Once that is done, then what you need to do is to just send this molecule to the panel of doctors, pharmaceutical specialists, and people, etc., who will say what is the dosage for the patient and what's the right thing to do. Is it safe, not safe, etc. Those kind of issues, and eventually gets administered to the patient, and patient is happy running, jogging, and all this stuff. So this is the overall pathway. I call this as a Dhanvantari pathway, and I'll tell you more about it. What does it involve? It involves genomics, proteomics, metabolomics, information technology, and definitely some chemistry, right? That's the dream plan. How do we do it? But before I do this, you would say, well, early in the morning, shub shub bolo, where we talking of disease, etc. Well, if you looked at the discovery of antibiotics, how it has been progressing. Now, all those useful penicillin, etc., all those streptomycin, things like that, amoxicillin that your mom would have given you when you were very small, like a baby, and so on. All those things are here on the chart. And the chart goes on till 1987. After that, there is a discovery void. What does this mean? This means that we thought we had come up with new antibacterial drugs and we have conquered bacteria. Therefore, there is no need for further discovery of the antibacterials. That is our feeling, our complacency. Right now, we are happy that we have overcome the bacteria problem. But unfortunately, bacteria started developing resistance, right? This AMR, antimicrobial resistance, so that's what WHO says, is a very dangerous thing. Now, if bacteria starts to become smarter, we have to become even smarter, start coming up with new molecules. But since we stopped it, and if bacteria takes over, then a simple fall from a bicycle can kill people because it can be a colony, right? It can be a colony for the growth of the bacteria. And since none of the existing drugs is working, so bacteria have they are going to take over the world, right? We don't want that kind of thing to happen. That would mean we have to restart our discovery programs, even for antibacterials, not only antivirals. Antivirals are a nuisance because they're mutating, and every time there is a new virus, we have to have a new drug molecule. Bacteria too, same story, right? So that's the reason why I'm showing flashing this. I've not put this antiviral. Maybe next time I'll probably add another slide on the antivirals. How do we go about doing drug discovery? Yeah, here are some milestones. Now, this was a Nobel in physiology medicine 1939 for Prontosil. This was an antibacterial, right? Do you remember this 39 to 45 was the Second World War time. So soldiers were dying due to various bacterial infections, and then people needed desperately some molecules. Prontosil was one of them, and then penicillin came a little later, and that is by Fleming and other co-workers, right? And then this this lady, of course, Hodgkin, solved the structure of the penicillin and found that it has a this four-membered ring, so-called beta-lactam ring. So once you have a structure of the drug molecule, you can start mutating, you can start making some modification, ethyl methyl substitutions, and ensure that you can beat the resistance. Some people are resistant to penicillin, so you can modify it and come with a new series of molecules. So that's where you need structures. And she also solved it, and she got the Nobel in 64. Right? And this was for streptomycin, again, a very useful, effective drug against tuberculosis. So that's 1952. So it looks like almost these are like serendipitous discoveries. By chance, things have happened and so on. So then you want to know, how do you go about doing things systematically? Of course, there was some such effort from these UU2, Omura, etc., who got the Nobel for physiology medicine, again for artemisinin, for malaria, etc. But these people suggested that there should be a rational approach, black, alien, and Hitching. And for that, they were given the 8 years. So what they said was focus on the pathway. Now, today we talk about drug target, right? And a pathway. And we want to inhibit that pathway. We want to inhibit that drug molecule. So that's more like a rational or structure-based drug approach. So this is a more modern approach to random screening of molecules and serendipitously discovering some new drug molecules. So you want a systematic way of going about things. And that's why I'm calling this how to design drugs. And this is what they suggested, that focus on the biochemical physiological process, right? So well, let us see how we utilize this part. In fact, I'm sure some of you recall Venki Ramakrishna's contribution and his Nobel in 2009. So that was for the ribosome. And what is so big about this? Well, it, of course, is a very big molecule for X-ray crystallography to solve this million atoms. It's a technical feat. It's a fantastic technical feat accomplished by these three scientists. But more importantly, it's ribosome of the bacteria are different from that of humans. So if you are able to block the ribosome using some small molecule, then you are eliminating the bacteria and the humans are safe, right? So that's the general idea of how to utilize structure of the proteins or structure of the drug targets to design drug molecules, right? So this is, in fact, Nobel citation does indicate that their discoveries paved the way for generation of new antibacterials, right? And why is it, in spite of with having so many millions of compounds, that we are not able to come up with a newer and newer molecule? So that's a big question that everybody would like to ask, right? Now, if you look at the number of molecules that have come out, in the last 80 years, or even you can even go backwards. I've gone up to 1940 here. You know, FDA approved drugs. FDA approved NMA stands for new molecular entities, which are on the way to becoming drug molecules, which are patentable, right? So only 2,500 drugs in last 80 years. That is amazing. In spite of having millions and millions of compounds, like half a billion compounds, and only this is not to say that there are only 2,500 diseases. Actually, there are very many more diseases than there are available drugs. There are some diseases where there are no drugs available. So apparently there is a problem with the drug discovery, and that too, in spite of increase in the genomic and proteomic data, the drug discovery pipeline, researchers are saying, is drying up. No new molecules are coming. Only modifications to the existing molecules are happening, not new molecules. So that's not very good news, especially with the latest genomic revolution, right? One aspect or one factor you can blame on is the cost and time involved in drug discovery. Now, remember the pandemic today, people are not talking of drug molecule. Instead, they're talking of vaccine. Let's get a vaccine. Let's get a vaccine. Two vaccines, right? Two shots. It's not a drug molecule. The reason being, well, drug molecule discovery seems like a very long process, 14 years and $1.4 billion revised to $2.6 billion, right? That's too much money and too much time. Of course, why does it take so much time? Most of it is because of the safety issues. So you want to make sure that the molecule that you create is safe to people. So that's why you have so many phase 1, 2, 3 clinical trials. That takes a very long time. Then there's review process. But I suppose if you in an emergency situation like this COVID pandemic, if you can come up with a molecule, I'm sure you can bypass a lot of these things. These days, now regulatory agencies and governments are very sensitive to this issue. They would like to make it as an experimental molecule. Probably it may not take this long and this much money. So there is a hope that if our in silico or computational solutions are reliable, and if you are able to generate that kind of a confidence in the community, in the regulatory agencies and in the government and in people, then probably we can cut down cost and time involved in drug discovery and start making molecules. We can realize our dream. Remember, we started with the 10 a.m. to 5 p.m. So in a day, within a day, we should be able to come up with a molecule, provided our methods are reliable enough that there is no toxicity and it's a highly, they go like magic bullet to the target and just disable only the target and not bother the rest of the system and so on, right? Let's see how we proceed with this. So this is what I think is happening now with this genomic proteomic situation. I'm sure there is some Kishore Kumar in the audience who can sing. Right? The current scenario is increasing population. Remember, every time there is a winter, there is a pollution in Delhi, and we're doing odd even, we're doing all kinds of things, increase risk of communicable disease. Obviously, this is definitely pandemic. I'm sure all of you are wearing masks, right? Please wear masks. Don't drop them, right? Increasing automobiles and so on. So many problems. So do we have a solution for all this? I'm sure we can find using this genomic proteomics. But currently, I will not talk of all those artificial plants to clean up the air, etc. Let's focus on drug discovery, right? And how do we do it? Our immediate plan is find new biomolecular drug targets and develop some new drugs. Keep some in store. Don't tell them you got them. don't expose them to microbes, so otherwise they develop resistance, right? So you have to have this plan of action. How do we do it? Well, this is a pathway I'm calling this. Remember, I was calling it as Dhanvantari, developed at SCO Bio. SCO Bio again stands for Supercomputing Facility for Bioinformatics at IIT Delhi, right? So this is our lab. What do we do? So we read the genome, and the plan is from genome, we identify the genes. This could be DNA or RNA based, so that's not an issue, right? Genome. And then we identify genes which are coding for proteins. And so this is our software called ChemGenome. It's a novel method which focuses on the language of DNA, oblique RNA, and to identify protein coding genes and other functional units on the genomic DNA. So once this is done, at the end of ChemGenome, what do you have? You have a bunch of genes on this genome. And now these genes, easy to write down the RNA part of it, because all you need is A to A, G to G, C to C, T to U. Instead of thymine, you have uracil, right? Instead of deoxyribose sugar, you have uh, ribose. So that's the only difference, right? And it's single stranded versus double stranded. So that's the other issue. But anyway, our focus is now on the basis. So from thymine, you go to uracil. That's the only difference from the 5 prime 3 prime direction. Now, once you have the mRNA, writing the protein sequence is easy. Again, I mentioned Hargobind Khorana's genetic code. 3 bases constitute 1 amino acid. So if you have a base sequence here on the RNA, what is this? This is serine, glutamate, alanine, arginine, threonine, etc. Single letter code for amino acids. So this, once you identify genes, going up to sequence is a trivial, very, very small high school level software, very simple software. After this is a question, big question. So how do you convert this protein sequence into a three-dimensional structure? Of course, you can do X-ray crystallography, you can do NMR, but they're time consuming. So can we utilize computational methods? So that's where this Bhagirath comes in. So Bhagirath software does the job of three-dimensional structure prediction, protein structure prediction. It gives you some candidate structures for the protein. Now, if there are sequences already similar to the given sequence in the protein data bank with known structures, that is, if the homology is high, it, the structures are reliable. If they are not high, if the homology is less than 30% and so on, I'll come back to that later on, a little later as we go along. Then, of course, you have to figure out how do we improve the accuracy of the protein structure. So this is a grand challenge problem, as all of you know. So sequence to structure is a big challenge, unsolved problem. So once you have the 3D structure of the target protein or DNA or RNA, some people now call microRNAs also as drug targets, right? So you could then utilize this small molecule development drug Sanjeevani software, which does the job of target-based drug discovery. Protein as well as right now it's useful for DNA as well as protein. We are working on the RNA part. So not what does the Sanjeevani do? Sanjeevani gives us some plausible molecules which can bind very tightly, inhibit the protein or the DNA sequence, and they can be tested further for their efficacy as drug molecules. So that's what this is the pathway is suggesting. Excuse me. Now, how does it work? Does it work at all? Right? You have a question. I also have a question. So, so my friend Professor Vivek Perumal is a world's expert on hepatitis B virus. He is in the Kusuma School of Biological Sciences, and he posed a challenge to me. He said, "Can you discover a new molecule for HPV?" He has all the experimental lab set up, so I have the computers and software. He says, "Give me a molecule. I will test it, whether it works or not." So well, let's see how we do it. But I asked him, what do we know about this already? He says something about this particular virus is known for ORFs. ORF stands for open reading frame, right? This coding for a protein. This also coding for a protein. And so this is something about the function of these proteins is already known. So he said this information is already available, but what you want to do is avoid polymerase, because people have utilized it already. Everybody is developing drug molecules against polymerase. Don't do that. And the other thing you should do is don't give me a nucleoside analog, because most of the drugs look like nucleosides. And what is a nucleoside, by the way? Well, AGCT, they are nucleic acids, right? When you add a sugar, they become nucleoside. when you add a phosphate, they become nucleotide, TIDE, right? So this is, so essentially this is like a sugar plus, sorry, base plus sugar, nucleoside analogs. And this virus has developed resistance towards this nucleoside analogs. So his challenge was, don't do polymerase as a target and don't make a nucleic acid nucleoside analog. So give me a new molecule for a new target. That was the challenge that he posed. I said, okay, we can mm, go to the NCBI. Remember, I told you this is free. NCBI is available freely. So all you have to do is go to your mobile phone or laptop, go to NCBI and type hepatitis B virus, and you will immediately get a genome, entire complete genome of this virus. So I take this genome, I download it onto my laptop, and I push it to my Chem Genome software 3.0. So this is one of the latest versions. So what does it do? It identifies 5 protein coding regions. So it does automatically via RNA. Remember, I was mentioning the DNA to RNA to protein, etc. So it gives you protein sequence, 5 protein coding sequences. I could have taken all the 5 because they don't share similarity with humans. So humans are safe if any one of them happens to be a drug target. But he said, don't focus on all of them. Just look at this gene 3, which is going for a surface protein and so on. All right, let's do that. Now, how do we do it? So here is my sequence coming from Chem Genome. What is this? Well, this is single letter code for the amino acid, right? Methionine. This is what? Glutamate, asparagine, isoleucine, threonine, serine, glycine, blah blah, etc. Goes all the way up to isoleucine here. So I again take this sequence, cut and paste to this Bagheera server. What does this Bagheera server do? Well, this H here stands for this hybrid, homology ab initio hybrid. So this is an improved version which can handle larger proteins and also incorporates homology as well as ab initio. That was this H business. In fact, my student now have another version called Bagheera H plus, right? That's another newer version to obtain this structure. So this Bhagirath gives me a 3D structure. Once I have a 3D structure, I start looking for pockets, binding pockets on this. Remember, I'm not using any experimental information. I started with the genome, went to this sequence gene. After gene, have a structure. Then I'm looking for binding pockets on it. So I identify about 10 binding pockets based on this ASF active site finder of my Sanjeevani software. Once I have the 10 binding pockets, because any one of them could be a potential binding site or active site. Besides, I'm not using the idea of active site right now because I'm not using any experimental information. There's a potential binding pockets. Now I go to the next module of Sanjeevani, that is RSPD, which is which has the ability to scan millions of compounds in minutes, which is the kind of thing that I want, right? And remember, we have so many millions of molecules. So why don't we use this data to see if any molecule can be used as a drug? But you don't want millions of years for this, right? So obviously you need a good program which can do the job. This RSPD of Sanjeevani can do the job for you. Right? And it's free though. Some of you who are desirous of doing drug discovery should try this RSPD Plus now, the latest one, which uses some additional artificial intelligence technique as well, in addition to physical-chemical methods. So coming back to this, so using this scanning millions of compounds in minutes, I shortlist about 150 molecules using some threshold value. After I get this 150 molecules, then I now go back to atomic level docking and scoring, use my PARDOC program of Sanjeevani. So what does this do? This puts all that van der Waals, electrostatics, solvation effects, etc. All those detailed atomic level calculations to obtain a free energy. And after that, I rank these molecules, all those 150 molecules, and I say, okay, maybe these top guys are looking good. So maybe we are ready to do the testing in the lab, in the laboratory, whether this really works or not. So then we took the top few molecules and then got them tested. This is molecule 1, molecule 2, 3, 4, 5, right? This is how the structures look like. Coming out of millions of molecules, remember? And now what does it do? Well, these are the contexts computationally, because if you're doing atomic level docking, you know what are the residues on the protein which are interacting with the drug molecule. So that's a listing here, and this is the listing. So my job is done. I said, I told this professor or IIT Vivek, he said, well, my job is done. In fact, he was asking me, how do we do it? Well, so it doesn't take 12 years and $2.6 billion. It just is free and it can be done in 3 hours. Right? We start at 10 o'clock. By 1 o'clock, you are ready with a list of molecules which can be tested in the laboratory. So it's as simple as that, right? Now, does it work? So Vivek finally did an experimental validation. This took a long time. In fact, a lot of journals, sent it back saying that, well, do this, do that, all kinds of tests. Well, we have done all of that stuff. Finally, what we found was that there was one molecule, molecule 5, which not only takes care of the virus, the wild type, as well as its mutants too, for which there are no drugs available, not even nucleoside analogs, right? So that was a good reason. And so this is the KD value, binding affinity. So all these experiments were done. So when I say it's a success story, I have the experimental validation. So what's my summary here? Summary is this: Dhanvantari seems to work. So a micromolar compound is routine when you're doing a micromolar. Nanomolar is what you would like if you want it to be drug-like to be sold in markets in the laboratory. So all that would mean is maybe we have to add one extra van der Waals contact here, maybe an extra hydrogen bond here. So you can do some little bit of tinkering here so that you improve the complementarity between the drug as well as the active site of the binding pocket. So that's feasible, right? But what is not that easily feasible is how do you ensure that it is not toxic to humans? This is where we are kind of stuck in this computational drug discovery. So now, if some of you in the audience can tell me these are the 5 rules of toxicity, then of course we can integrate it, create a software, and then say that, well, this molecule within 3 hours you can do this, and this is going to work, will do wonders, and it's like a magic drug, right? And you can administer it to the patient. So that toxicity part is still not amenable to simple manipulation in terms of rules, right? But so what do we do? Well, we have to hypothesize. Well, what is toxicity due to? Is it because the molecule is breaking metabolism? The metabolizing enzymes are breaking it, and the fragments are no good for a drug molecule. They are blocking, they are spoiling the biochemical pathways. Or is it that they are going to wrong sites, off target binding? So you can make a proposal, you can make a hypothesis and test it computationally, which is what we are doing now. But it would be ideal if somebody like Lipinski rules, somebody can come up with some 5 rules of toxicity, so we can screen the drug molecules, right? So that's where the future of drug discovery is. So automated drug discovery is feasible, nanomolar compounds, etc. All this is feasible, but what we have to do is conquer that toxicity part. So that's a summary of this genome to hit molecule. So now I'm going to give you more in details, but essentially you got the picture. You know what I'm talking about, right? Okay. What is chem genome and what is a genome annotation model, and why do we have to do a genome annotation at all? So a little bit of history for you. The history is first viral genome, 1976. Bacterial genome, 95. Eukaryotic, and everybody knows this as Craig Venters, Celera, and NIH joining together and giving this gigabases, right? This is a human genome, right? And how does it look like? So the genome looks like CAT, GAC, GTG, CC, etc. This is what the genome looks like. Earlier, it was taking 15 years and $2.7 billion, and now, of course, you're talking of a single day to do the whole genome and less than 1,000. This is coming down every day. So this is gives the hopes that one day we will have genome card for everybody. Right, very soon, all of us will carry that genome card, right? Which is a very unique, even more unique than other card, right? And what is interesting is that this person, Frederick Sanger, was responsible for the DNA sequence, and he got the Nobel in 1980. This is 1980, but he also did the protein sequencing in 1955. So he was one of those few who got 2 Nobel prizes, one for protein sequencing in 1958 and DNA sequencing in 1980. So he paved the way for today's genomic sequencing efforts, right? So that was his major contribution. Now, on the genome part, why are these various organisms different? The humans versus plants and viruses, etc. We put ourselves at the top of this evolutionary tree. We are somewhere out here, and all because of the genomic diversity. So, if you understand genome, well, we understand it all, right? That's basically our our proposal or a hypothesis. So whether it's going to be Tendulkar or Sindhu, etc., Mitali, well, all in the genome, right? And it's all in the genome. Of course, there will be environmental heredity versus environment. All of you will agree, right? I've been saying something about DNA to protein and drug molecule, etc. Well, Oh, I'm assuming that all of you are aware of the central dogma of life, which is genome, transcription, RNA, translation, protein, and 3D structure. And this is our drug target, right? Most of the time. Proteins are the nanobio machines which carry out the functions that keep us alive. So we are focusing on this part, right? Now the question for you, for the audience, this is a question for the audience. So I'm now going to flash two sequences, a red one as here as well as the blue one. CCA, GGA, TGT, TD, etc. So what is the meaning of this thing? Can you read this? Now, if it was Twinkle, Twinkle, Little Star, everybody understands it, right? Even kids understand it. But this I don't understand. GG, GT, TT, what does it mean? Unless I give you the definition that, well, this happens to be that handsome boy, this happens to be that naughty girl, right? So that unless I tell you, and how do I tell you? Well, if I already know from the database, using homology, using some comparison, using a blast, etc., then I can tell you, well, this is coming from chromosome X, this is from chromosome Y, because there is a match with something that's already known. Suppose there was nothing known, then you wouldn't have been able to predict it. This is what I'm calling as the language of DNA. Nobody seems to understand the language of DNA today, except probably one person. My suspicion is this man knows everything. He's not talking, but I guess he understands it all, right? So if you want to solve a big problem, focus on the language of DNA, apart from that protein folding problem, which I'll come to very shortly. So in this genome to drug, we have three major challenges, and it depends on how much time you have to to solve in your lifetime to solve this problem. One is language of DNA, second is protein folding, third is get rid of toxicity in the drug molecules. Then you have solved three fundamental problems so bothering society, and you will advance science tremendously in a humongous way. Okay, now coming back to the gene prediction softwares. So in the absence of understanding of the language of DNA, what do we do? Well, we write softwares with something known already. We train the software using some limited experimental data and then start to make predictions. So we are doing training and testing. So we train the data and then we do a testing. So most of the softwares are like that. You know, ab initio, they do write ab initio homology, but then they are doing mostly training, right? Because chem genome is somewhere out here in one of those ab initio methods. What is the problem with most of the software? The software seem to be trained for a particular organism, meaning that from that organism, with a limited experimental data, you will train your model and you'll make predictions on the remaining part. Suppose there are 5 genes known experimentally, you'll train your data on those 5, and you try to predict the remaining 95, right? So of the 100 genes. So challenge is that if you trained it on one organism, your accuracies may be like 97%, 95%, etc. You go to another organism, suddenly same software starts to fail. No, you'll eventually come away some 30%, 39%, 5%. Terrible. This is terrible, right? And sensitivity is, your accuracy drops if you cross the organism, if you go to another organism. That's the major challenge people are facing. So especially with the availability of the new next gen sequencing methodologies, the NGS technologies, etc. What are the challenges? Challenges are, how do you assemble a genome? Then how do you annotate it? How do you understand what's the role of each and every base pair, right? If these two are done, then you have solved this major problem in understanding the language of DNA, or in understanding the genomic organization, right? And you will also be helping the NGS people, you'll be helping the DNA people, everybody, right? So that's the goal here. There is no universally applicable model because these softwares are organism dependent. If you train the software for a virus, it doesn't work for bacteria. If it works for bacterium, it doesn't work for a plant. If it works for a plant, it doesn't work for human. So this is not good. Watson and Crick did not do it. Remember, Watson and Crick said A pairs with T, G pairs with C, no matter what the organism is, virus, bacterium, human, plant, irrespective of it. So they have a universal model. We don't have a universal model. Now, once again, to highlight that point, here is one organism, Arabidopsis thaliana. Some softwares here. Now, sensitivities, don't worry too much because people are continuously training, coming up with better and better sensitivity over the years. So as you can see, there is room for improvement. It's not 100%, right? It's not 1.0. 1.0 here. No, there is it's less than that. That means there is room for discovery. So some of you can contribute. No, let's do more research here to make sure that it is 100% accurate. But remember what is going on with the methodology. This is more important to us. So this is fifth order Markov model, discriminant analysis, pattern recognition, neural network. So very sophisticated mathematical statistical techniques are being utilized to do the gene prediction. Some of you, if you recall, on the human genome, people said there are 20,000 genes on the human genome. This was Craig Venter. What were they using? They were using a higher order Markov model, 11th order Markov model, 15th order Markov model, that kind of thing. So they were all using mathematical. So this, what do you do in these methods? Well, you train these mathematical models on the existing and the known experimental data, and then you do a prediction on the unknown part. Now, your model becomes better and better if you have larger experimental data, more experimental data. But if you have 100% data, then there is no need for a prediction, right? So what we need is not a Markov model, but something that's universal, something that works without training, right? Remember, A pairs with T, it doesn't have to be predicted by a Markov model, right? You should be able to say based on chemistry, based on hydrogen bonding, A and T make 2 hydrogen bonds, G and C make 3 hydrogen bonds. So this is, there is no pattern recognition involved here, right? This is simple chemistry, complementarity of hydrogen bonding. If you can capture that type of a principle and then utilize your mathematics and statistics, then you'll be a success story, great success story, right? So what I want you to do is to look at the genomic data and look at the genes and figure out, well, what are the principles here of recognition or principles of, of this gene expression. So that will be wonderful if you can do that. So, but all said and done, if you are looking for a universal model, you cannot ignore the chemical nature of bases, because Watson and Crick did not ignore it. In fact, they utilized it successfully. I'm sure you also will be able to do it. So this is adenine, thymine. This is a double ring, single ring. I call it as a purine. This is a pyrimidine. This is a purine guanine, and this is a pyrimidine cytosine. G pairs with C with 3 hydrogen bonds. A pairs with T with 2 hydrogen bonds. Right? Remember, this is what is shown here. C and G here. A and T. A and T. A T G C T A. So this is a pairing, base pairing. Now, what is this part? I'm calling this as pairing, G and C, between these two strands. This part is a stacking, how one is stacking upon the one pair is stacking on the other, one pair on the other, etc. Right? So that's the stacking part. This is the pairing part. Now, let's move on to the next issue. Suppose now you want to develop a universal model and you want to utilize this idea of base pairing, stacking, like Watson and Crick, right? And what somebody has to give you these numbers, hydrogen bond, stacking energy, and so on. And I'm including one more parameter here, third parameter, which is what proteins do. Proteins are always looking at the grooves of the DNA. Remember, what is a groove here? Well, here is the groove here. So this part is all the groove here. There's another groove here, groove here, groove here. So these are all the, and proteins are coming and binding from these grooves. They are making hydrogen bonds. They're making nice contacts, van der Waals contact. They're looking at phosphates. They're doing all kinds of things. Proteins know how to recognize DNA. We don't know. That's a problem, right? So let's say there is a third parameter. In the absence of protein, solvent water is doing filling up all the grooves here, right? So we can call this a solvation energy or protein nucleic acid parameter. So basically, we have 3 parameter model: hydrogen bond, that is base, one base interacting with other base on the other strand, and stacking, that is going vertically here in the 5' to 3' direction. One pair interacting with another pair upwards or downwards like a ladder, right? And the third is the grooves part. Now, utilizing these three, let's not worry about viral genome or bacterial genome, plant genome, human genome. Let's just think about bases, only DNA and its bases. So think DNA, not the organism. That's what universal model is supposed to do. Now you need numbers. Well, these numbers can come from so different sources. One, of course, is your smartness, your uh, paper pencil, brilliance of the brain, and because AT pairs with 2 hydrogen bonds, GC pairs with 3 hydrogen bonds, you can easily say, well, maybe this is a number. Alternatively, you can do a molecular dynamics simulation. You can take DNA of all sorts of sequences. You can do long molecular dynamics simulations. That is solve F equals to MA on the computer. Newton's equations of motion on the computer for a given DNA sequence, and you can pull out, you can do analysis of the trajectory, you can pull out this. So these things have come out of MD simulation, this data from our laboratory. So we have utilized the MD simulations on long MD simulations on various sequences, and we compile this. Now using this lookup table is very easy for you. I give you a sequence. So all you need is a paper, pencil, or your laptop, or an Excel sheet. And then, well, if a sequence has A, then I'll say A this much, AC this, AG this, etc. So I can just, if you have the nucleotide, I can add up all these numbers, right? So that's very easy for you. And then if I give a long sequence, you can say this is a cumulative hydrogen bond, and you can divide it by the number of bases and say hydrogen bond per base pair, or hydrogen bond stacking per base pair. So that's what you can do once you have the table in front of you. So I'll skip this part for now because this is a long story, except that those of you who have seen this type of a table in Hargobind Khurana's, three bases giving you one amino acid. Here I'm putting the DNA code, not the RNA. In the typical textbooks, you will see instead of thymine, you'll see uracil, UUU, right? UUC, etc. Here it's TTT. So, and there is degeneracy. There are 64 codons. Here, of course, in the DNA space, you don't call them codons. RNA space, you call them codons. In the DNA space, you'll call them as double helical trinucleotides, right? The interesting thing is there are only 20 amino acids where there are 64. Now people blame it on wobble. There is no Watson, Crick, of course, not Watson. Crick considered this issue. He said this is because of the wobble. Third base wobbles and therefore you have less number. Now if third base is unimportant, then you should get only 16 amino acids. Either it's 4 cube or it's 4 square, not somewhere in between. But we have 20 amino acids. Right? How do we account for this degeneracy? Wobble doesn't explain. So we are asking what is the molecular basis of wobble? Now, if you go through this slide and probably this or any of my presentations, you'll see that stacking and hydrogen bond can explain the degeneracy, which is why I've come to this idea that you should focus on hydrogen bond stacking if you want a universal model, right? Now, how do we now go about doing this universal model? Well, so give me a sequence. I'm going to add up hydrogen bond in a given direction, 5 prime to 3 prime, then stacking and solvation. And then I convert that into a simple vector. This is simple basic mathematics, right? So I convert them into a resultant vector, three-dimensional resultant vector, and I'm asking what is the orientation of the vector? If there is a difference in the composition or in this energetics of the gene versus non-gene, I should see a difference in the orientation of the vector. That is the hypothesis. Does it work? Well, it seems to work on 900 prokaryotic genomes. I didn't have to do any training. The blue ones are all, each one is a gene here. So there are lots of genes. So this is one organism. This is another organism. Now each organism has several genes. So each point is that. Now what is a non-gene here? Non-gene is a frame shifted gene. You frame shift it by one base, then you get lots of stop codons, etc. It doesn't code for any protein. So these are all protein coding DNA sequences, whereas these ones are not coding for anything. So you'll see a nice separation in various organisms, not only just one organism, and you didn't have to do any training. So this is what I'm calling as molecular dynamics based physicochemical model for gene prediction in prokaryotic genomes. We extend it to 2000 prokaryotic genomes and it works every time, right? And then once you have the idea, writing a software is an easy part. In fact, in my group, in my laboratory, one person, of course they're all working with me, one person develops the idea, the other person writes a software, then another person writes a web server. So you have a software, you have a web server. So 3 4 people, there is a bioinformatician, there is a chemistry person, there is a biology person, right? And there is an IT person. So these 4 people join together to create a nice software. So it's a teamwork, and you need teamwork these days, right? So we have to promote more and more of this teamwork in India among our Indian students. Then you'll be a great success story. So writing a software is easy, right? Read the genome, 6 reading frames, etc. Then look for these 2 RFs, find out the resultant vector and see whether it's above the plane or below the plane. If it's above the plane, call it a gene, and then you have you can put some additional filters to remove the false positives, etc. So that's the chem genome software. Of course, so you don't have to write a software though these days. Now chem genome is already available to you. All you do is cut and paste from NCBI, get the sequence and cut and paste in this box, and then submit. Just click the button, and then if you give a email, then the answers will come to you. Otherwise, you have to look look for your job ID and look for the answer and so on. So that's how it works, right? Now let's go back to that previous slide that I showed you earlier and ask you, well, where does it fall? Well, it shows up somewhere here: sensitivity, specificity 0.87, 0.89, compared to the other sequences. And luckily, this model is nearly universal. It applies to various organisms, not just one organism. Not only for prokaryotes, even for eukaryotes, as you will see in the subsequent slides. Now here I'm looking for promoters versus gene met A, met B. Now promoter melts at a lower temperature, and now my hydrogen bond stacking energy can capture this. So I can easily detect the promoters, not only genes, I can also do the promoters. And here is the next slide from rice genome. Now rice, remember, rice is eukaryote organism, not prokaryote, right? It has introns, it has exons. These introns are the black ones here, exons are the red ones here. But note this: if you're doing melting temperature or hydrogen bond and stacking, you get these little valleys here and the peaks here. Peaks correspond to exons, valleys correspond to introns. So they get spliced out, right? So it's easy to detect introns and exons utilizing this type of methodologies, right? More interestingly, this is a story of 7.6 million RNA genes, and these RNA genes are coming from mRNA, tRNA, microRNA, small nuclear RNA, ribosomal RNA of different kinds, right? There is some big one, small one, etc. Different types of various types of RNAs. And what happens when you look at the physicochemical properties of these RNA sequences? So here is a hydrogen bond. This is a solvation. Here is a stacking part. All of them are clustering at different parts on this three-dimensional space, right? So that clearly says that they are separable. So I will call this as DNA is talking. Of course, this was got published in 2016. I call this as physicochemical fingerprinting of RNA genes, right? Right. So this is an interesting story to tell, right? More interestingly, promoters. There is too much promiscuity in these promoter sequences. Strong promoters, weak promoters, but there is no consensus. So using sequences, you cannot detect, well, is this guy going to be a promoter or not? Very difficult to figure this out. Now, if you, for example, yeah, all the sequences are doing the same job and they're coming from different organisms. If the base is conserved, you will see same color and or the same position. But as you can see, there is no one single column with the same color all across. So there's too much variation, although the function is the same, right? And how do we then solve this promoter prediction problem? Now, all you have to do is to go to these physicochemical properties and then scan the sequences. What you see is either a peak or a valley at that promoter site. Wherever there's a promoter, there is a nice signature. So that's what is captured by these physicochemical properties. So I believe DNA is talking, ready to talk. So just that we have to tune in to that FM 98.3, right? Which we are not doing. We are busy training, not thinking about DNA. So the message for today with this part of the lecture is think DNA. Intron exon I mentioned to you already. So you can see that there's a nice intron start site. There's a little peak here. There's a nice signal here for the intron end. So this is a good story from eukaryote points. And remember, intron exon, there are just two bases on either side, and it's difficult to train this, which is very difficult to train. Sequence is not conserved, but physicochemical properties are conserved, however, of the sequences. So that's where we have to move towards the sequence and decipher the properties which are relevant. Now that we have these properties figured out, we can now use these artificial intelligence techniques and the machine learning methodologies to come up with better softwares. So this got published very recently, 2021 NAR. NAR is the Nucleic Acid Research Journal. Right? This is my PhD student Akhilesh Mishra, and that's the story of the part. Now the message for genomic part is someday we should read human genome like a Harry Potter novel. That's my dream plan. Rice genome is novel too, Buffalo. There's so many novels out there in NCBA. We don't seem to understand. We are missing all the fun, right? And I've been focusing only on the coding part. That is this 3%, which is uh, an ATMB. Now, what about this non-coding, all these other things? And so there's so much to learn from the genome. Some people say that only coding is relevant, rest of it is redundant. What is it redundant? What is this? And they call it as junk DNA. How can it be junk? If it is junk, we would have discarded it a long time ago, right? Evolution would have discarded it. Maybe that is incipient superhuman DNA, right? Some of you may be already on the way to becoming superhumans. So who knows? You are the next generation people. So you may have that incipient DNA with you. This extra DNA may be doing that job. So it's a interesting Harry Potter novel that we got to decipher. So let's understand the language of DNA. But now move on to the protein story. DNA part is done. Now, if you ask me a very simple question, why do people fall sick, right? What is a disease or a disorder due to? A very simple question, right? Well, it says either protein going crazy, is either overactive or is underactive. If it's overactive, well, you have to come up with a molecule which will slow it down. If it's underactive, then you need an activator. Most of the drug molecules in the market are inhibitors, not activators, right? Because there are other methods, not only protein targeted stuff. And I was mentioning to you that DNA also could be a nice target, but because of the drug delivery issues and the size of the molecules, we have some hesitation, reservations on DNA becoming a popular target. But if with advances in nanobiotechnology, I believe DNA also could be a very popular drug target someday soon, right? So that's the introduction to this structure-based drug discovery, right? And why do we, what is then bothering us? Why can't we just go ahead do it? But our protein data bank doesn't have all the structures. This is a basic problem. See, out of 20,000 plus proteins in the human, only 7,000 have structure. And I'll show you more data in the next slide. And similarly with the other organisms, and your sequences are going in millions, structures are happening in thousands. So there is a big, huge gap between these two. So we don't have enough structures, right? Another simple numbers here to understand. If you want to cure malaria, that is Plasmodium falciparum, right? So we have 5,000 sequences in the, this is a Vivax falciparum strain, this is Vivax, and Vivax is the more bothersome for Indians. And now 5,000, only 53 structures. So how do you do drug discovery? It would be nice if you had all the 5,000, but then it takes too much time using X-ray crystallography, NMR, cryo-electron microscopy, etc. So it would be nice if you have a computational method, right, to come up with structures, reliable structures. So this is calling for essentially a computational protein data bank. We need to do something about this very quickly so that we can start solving this. This is a tuberculosis, so 4,000, only 380 structures. Human, no, I just mentioned 7,000. This is a rice genome. You want to know more about rice? 24 structures out of 28,000 sequences. This is no good. Right? These are gene sequences, are protein sequences, and some story with other chikungunya and other stuff as well, right? So why don't we then predict the structure? What is the big problem? Well, I'm sure some of you know this protein sequence. If you start looking at the structure, this is a backbone of sequence, and R here refers to the side chain, that is the amino acid, and this is a common backbone for all the amino acids, right? So this is CONH peptide bond, right? And here is a phi, there's a psi. These are the dihedral angles, torsional angles, and these are called Ramachandran angles. So if you can specify the Ramachandran angle phi and psi around each amino acid, you solve the problem. That's it. All you have to do is just mention phi psi. If this is amino acid, well, this is a phi and psi. Then that's it. You've solved a grand challenge problem pending from 1950s. Remember, Pauling solved the secondary structure alpha helix beta sheet in 1952. After that, what else has happened? 1959, Kaufman said oil and water don't mix. And after that, anything else? No, nothing happened after that. 1959, 2012, 2021, no solution to the problem. Best of the brains from Harvard, Cambridge, everywhere, right? They're all working on it, and we still have to see a solution to the problem soon. So all you need is just specify phi and psi for a given R, and you have it under control. And all the diversity of the structure C is because of this phi and psi, Ramachandran angles, right? Simple. And additionally, what else do we know? Einstein said that this protein folds and folds and folds, and eventually from a sequence, eventually goes to settles at the bottom of this well. This is a free energy well. So it is characterized by the minimum free energy, right? So this is a grand challenge problem. in life sciences, in physical sciences, and computer science. Now, why computer science? Well, this is a multi-dimensional parameter problem, and you're saying optimize, and you have a cost function. In this case, it's a free energy function. So you're optimizing the cost. You're trying to say, give me a combination of the parameters. It's like, it's like that combination for your digital lock, the zeros and ones, which gives you, which opens your box, or which here gives you the minimum numbers. It's almost like, let's say you have some 100 different ways of incomes and expenditure. You want to know what is the combination of the expenditure and interest, which will maximize my savings, right? There's a problem in economics, right? And that's an unsolved problem. And mathematicians call these problems as NP complete or NP hard problems. So protein folding is one of those NP hard problems, right? So it's a grand show. If you're looking for, I want to kick in life, I want to solve something fundamental, well, here is your chance. Take that simple question. What is the phi and psi for a given hour? Then you have solved it, right? And obviously it'll automatically become the minimum in the free energy here in this well. So suppose you want to use these supercomputers to solve this problem. Why not do F equals to ma for Newton's equations for a protein? In fact, this person called David Shaw created Anton machine. This Anton is a super duper computer dedicated for protein folding. So what it does is, of course, the F equals to ma is coded on the chips, and so in that sense it's extremely powerful. And now it can do F equals to ma for 50 amino acid. 200 is too big. 200 it'll take to 10 to the 3 years, 1000 GPUs 1 year, etc. So he can do it within 1 month, right? For a 200 amino acid. 1 month is too long. Remember, I want a drug molecule by the end of the day. 10 a.m. I have reported. 5 p.m. I want the drug. I don't care, what you do, GPU, CPU, etc. Right? You will insist like that. Obviously, this is not good enough. Anton is nice, good to have, but not good enough. Alternatively, you can generate millions and billions of configurations. Suppose there are 200 amino acids and 2 degrees of freedom, that pi psi, right? And let us say they are taking lots of values, but here we focus on only 2 values. So you have to have 2 to the power of 200 structures. That is, even if you take 1 minute for each structure to calculate free energy, you're talking of so many 10 to the 54 years. It's an impossible thing, right? So this is why you're calling it as a grand challenge problem. Okay, but what does Bhagirath do? But this is a Bhagirath pathway. I call it Bhagirath because Bhagirath did the impossible of bringing holy Ganges from heaven to earth, right? That's what he has done. So it's like as difficult as that. So sequence to structure is the issue. How do we do it? Well, we start with amino acid sequence, predict secondary structure, then do loop sampling and generate some biophysical filters, and eventually scan them for energies and report native like structures at the end of the day, right? So this is how it looks like. Now, instead of writing, so here is a sequence. So you preform the secondary structures, you bend them at every which way. Here, the loop regions, and then you have several trial structures. Here in this case, for this, you have 163,000, sorry, 16,000. And then you scan these energies, calculate the energies, and you superpose it. How good are you? Here, the blue is a native meaning X-ray structure. X-ray structure, red is a predicted computational structure. So this, what it is giving us is a confidence that you can get a structure within this RMSD. RMSD meaning root mean square deviation, 4 angstrom. So if 0 RMSD is obtained, that means it's superposing perfectly. If the atoms are deviating a little bit from the crystal, then there is a residual RMSD, 4 angstrom. 4 to 5 is what is achievable. So energy rank is 4. Here, of course, there is a better structure, 2.87, but its energy rank is 1000, so you may discard it. So the idea was, if you pulled out top 5 or top 10 structures or top 100 structures, do you have a structure in this basket which is close to the native? That was the question, right? So this is apparently yes. The answer is, of course, we have done it on several of them, 80 small globular proteins, and every time we are able to get a structure, 5 angstrom structure, right, for smaller proteins. We call this as the ab initio method. Now, after ab initio, we've said, well, let's just make an implementation on a supercomputer, because you have several structures and you have to calculate energies. This is a heavily compute intensive. So it's a 3 teraflop machine, and it's at 280 processors. So the user inputs amino acid sequence, then it does all the secondary structure prediction, extended structure, trial structure. So each of these yellow boxes is for one processor. So you're using lots of processors here, 280 processors, remember. And then eventually all these processors are they're sending their predictions to the master node, which does the energy ranking, structure evaluation, and finally 5 candidate structures. That's the so-called ab initio uh, overall framework. This we did this in 2006, but ever since we've been fine tuning, etc. So after that, why not tackle proteins of any size and fold? That was the motivation. So we added one H here, which is a homology. So combine homology where it is available. So you have a Bhagirat ab initio part, but if there is homology available, then utilize that homology. Then you come up with an output which has a larger decoys, which probably contains a native like structure. So that was the idea behind this H methodology. Now, ask one simple question. Now, if you say protein folding, well, it's unsolved. Protein DNA recognition, which is involved in, remember, gene expression, because whatever DNA does, it just codes, it has the information. Who is expressing the information? It's a protein which is facilitating expression of the information contained on the DNA, right? So that's a DNA recognition. It's an unsolved problem. Protein RNA, unsolved. Protein, unsolved. What is a common factor to all these unsolved grand challenge problems in biochemistry? Protein, probably. Then you start asking, are we looking at proteins right? Is there something going wrong here? Why is that so many problems related to proteins are unsolved, including drug discovery? Most 90% of the drug targets are protein, and drug discovery is about small molecule binding to a protein, right? And that's a recognition problem again. So all of them are recognition problems, and we don't seem to understand what's going on with these recognition problems. Now, what do we know, however? Non-covalent recognition beyond hydrogen bond. This is Watson and Crick, because they solved A pairs with T, G pairs with C. Using that, they solved the single strand recognition problem in DNA, right? Watson Crick solved that problem. And what did Linus Pauling do? Well, Linus Pauling said that some stretches of the protein sequence become alpha helices or beta strands, etc. So he was proposing the backbone hydrogen bond, CONH backbone hydrogen, I2, I plus 4, etc. So that was a again recognition problem based on hydrogen bonding. This is also hydrogen bonding. Now, beyond hydrogen bonds, have we solved anything in recognition related to biomolecules? We have a lot of proposals, but no, probably not. We have not solved it. Textbook classifications. Textbooks are classifying these 20 amino acids in every which way, so many different ways: aliphatic, hydroxyl, aromatic, basic, acidic, amide, sulfur, charged. I don't understand why they are classifying in so many different ways, but they are doing it, right? And the question that I'm asking is, do we understand the amino acids properly? Is there a problem figuring out the proteins? Well, what do we know so far? Well, we know, according to Einstein, sequence has all the information to give structure. This part is known. Everybody knows, and he got the Nobel for this in 1972. What else do we know? Well, Linus Pauling said that alpha helices and beta strands form, beta sheets, etc. They form based on hydrogen bonds. And the crystal structures are demonstrating myoglobin, hemoglobin initially. Further on, now several of them. These are easily visible in 3D pictures. So this is a Nobel Prize in 1954. Right. By the way, he also got another 2 Nobel Prizes. So apart from Sanger, Linus Pauling also got 2 Nobel. Right. Say Sanger, what did Sanger get it for? Protein sequencing and DNA sequencing. Linus Pauling got it for 2. There are 2 other people, by the way, who also got 2 Nobels. I'm sure some of you know in the audience. Okay, what else do we know about proteins? Hydrophobicity, yes, meaning that oil and water don't mix. So people have taken this idea to a structural level interpretation, meaning that hydrophilic residues, people, that residues which are liking water, which have hydrogen bonding potential or charges, or they get exposed to water, and hydrophobic non-polar residues like alanine, leucine, isoleucine, etc., they will be buried inside. That is a structural interpretation of a thermodynamic concept. The hydrophobic cities thermodynamic, right, in nature. But we give a structural interpretation. Now, what about Ramachandran's plot? What Ramachandran said was that if you plotted that phi psi, remember we said all you have to do is specify phi and psi, you solve the problem. In fact, Ramachandran helps us saying that not everything, phi can vary from -180 to +180, like 360 degrees. Psi also can vary 360 degrees. Not everything is occupied. Only this little portion, 15%, that's all that is occupied. So if you can figure out phi and psi in this 15% region, that is these dotted areas, phi and psi, then you have conquered the protein folding problem. Unfortunately, these four ideas I mentioned to you. What are the four ideas? Sequential structure by Anfinson, Pauling's secondary structure, Walter Cosman's hydrophobicity, and the 15% exclusion or excluded volume due to steric clashes. So these are not sufficient to build the three-dimensional structures of the protein. We still have millions and millions of structures to cope up with. So that is where we have the issue. Of course, with regard to Ramachandran, if you can use higher order Ramachandran maps and bring it down to 2%, 15% is not sufficient. 2% is what is happening. And then you can probably predict the 3D structures of small proteins. That we discovered. One of my students, PhD student, Debarati, she did it in 2015, right? That she thinks that protein folding is a convergent problem. It's not a NP hard problem, etc. Right? Now, I have another interesting story for you to think about today. Now, that story is, suppose, now you ask this very simple question. I'm sure some of you, have some very small, tiny little kids who are asking all kinds of embarrassing questions, and finally, mom says, forget it, man. Just go, just go play. Don't ask me any more questions, kind of thing. So I ask you a very simple question: why only 20 amino acids? No, I look for an answer in various biochemistry books and biology books, etc. There is no answer. They blame it on evolution, and that is a very favorite pastime. If you don't understand something, we say, oh, evolution somehow. Evolution has an answer to this kind of thing. Now, suppose you look at this triangle, equilateral triangle, all sides are equal, 60-degree angle. Use four colors, or red, blue, purple, green, or red, blue, same colors. How many color triangles can you make? Four, four here. So it's going to be 4 cubes. So it's going to be 64. But actually, because you can rotate it triangle by 120 degrees, if you don't label the sides, then they're all equivalent, right? If you rotated it, and similarly, if you flipped it by 180 degrees here, right, so it will be the same story. So how many unique triangles are there? Well, actually, they're going to be only 20 unique triangles. Now, how? Well, one combination is all three sides are red. One second is one side is blue, next side is purple, next side is green, like that. If you start labeling them, only 20. Now, what is interesting, however, is that any color, if you're using this, any color occurs in exactly 10 triangles. Red color occurs in 10 triangle, blue, purple, green, etc. Now, I am saying, I'm bringing this up because of the importance that this mathematics carries. So this is the mathematics of a triangle, equilateral triangle. Any two colors occurs only together in 4. Any three colors, because there are only 3 sides, only 1. Now, if we were to say, if I were to say, dare say, right, that maybe amino acids have, there is, it's just not mad evolutionary thing going on. Maybe there is a systematic way of building the templates. Maybe there is a logic, a rhyme and reason behind why 20 amino acids. Probably amino acids have evolved out of 4 properties. Each property, and 3 properties are used at a time in each amino acid, right? Suppose you were to say something like that. If you can always speculate, but then you have to satisfy all these conditions. If you are using 4 properties, then these 4 properties must occur in 10 amino acids like this, and any 2 properties should be occurring in 4 amino acids. So you have severe restrictions. If you want to appeal to mathematics, then you have a problem, serious problem, that you have to obey the rules of the game, right? So let us see how many rules do we come up with. Amino acid chains have evolved out of 4 properties. Each property occurs exactly 10 amino acids. Any 2 properties occur simultaneously, only 4, and so on. Now, what about textbook? Let's go back to the textbook. Leninger, Strayer, whatever your favorite textbook is. None of the textbook classifications satisfies the above rules. This is terrible, right? And either these rules are irrelevant, or probably they're all wrong. All the textbooks are wrong. Maybe we have to redefine the amino acids, right? Probably the rater is right, because we have not solved the protein folding problem. We have not solved the protein recognition. Maybe we have to rethink about amino acid. Now, Let us see, does it really work? Now, if you go back to basic chemistry, organic chemistry, high school organic chemistry, sp3 hybridized gamma carbon, I'm sure all of you know. Backbone has a C alpha. First carbon away from backbone is C beta. Second carbon is a C gamma. sp3 is saturated, and you have sp2, sp hybridization. So how many amino acids have an sp3 gamma carbon? There are exactly 10. How many amino acids can donate a hydrogen bond? Exactly 10. Do these guys occur only 4 times together? If you see, well, these are 4, here is 4. What are common? There are exactly 4 residues which are common to these two properties. How many amino acids don't have a delta carbon? Exactly 10. And what about these two? Well, there are only 4. What about these, the 1 and 3? Only 4. How many are have absence of branching? The fourth property, right? How amino acids have absence of branching. Again, you find that there are 10 of them, and all these rules are satisfied. Interestingly, none of this is mentioned in any textbook. So now you can see why people don't explain why 20 amino acids. So I'm calling this as a chemical logic based modeling. Now let's go back to protein structure modeling. So utilizing this, I now do this so-called regular blast methodology. Remember, idea of blast is to detect similar sequences. So I put an amino acid sequence, and then I'm going to use this similarity in the space of these properties, 4 properties, not in the space of letters, single letters. In the space of these 4 properties, I start detecting things that don't look similar at the sequence level. Just like the physical chemical properties of DNA, if you look at sequences, you don't find it. If you look at physical chemical properties, you will find their signals are very clear. Something similar happens with the amino acids too. And then now you can do a very nice homology modeling. So this hybrid method is integrated now with the ab initio. And this actually sets a higher watermark threshold for homology modeling, and it unravels the hidden similarities in sequences. So that's the advantage of it. And of course, you have to have a validation scoring function. So that is a Bhagiradesh pathway now. Now, there is an ab initio part, there is a new chemical logic, and this is a higher order Ramachandran map. So that's our Bhagirath H plus. So it has 5 candidate structures and so on. Now, how good is this method? Well, there is a competition called CASP. CASP is a world competition, critical assessment of structure prediction, right? And this is held once in 2 years, and a lot of servers participate from all over the world. And from India, of course, there is only Bhagirath. Nobody else is participating. Right. What you found was this 2016 official page puts Bhagirath at H plus at the 11th position. I was not very happy to see 11. I wanted to be a number 1 or number 2 here, right? Well, if you analyzed it carefully, you'll find that it is actually at number 3 position, right? In this analysis in terms of RMSD or GDT or whichever unit you take for analysis, right? That's the interesting part. And similarly, in the next CASP 13, they put us at the 13th position. Bhagirath Star was at the 13th position. Once again, something similar was happening with 13 too. When you focus on the RMSD, uh, there's a number of targets predicted under 5 angstrom RMSD. Bhagirath is somewhere out here. But I think these organizers have a very different way of evaluating things in any case. Last year, of course, was a disaster. This is due to pandemic. The team was not available. So hopefully next year we'll get to the top here and wave a flag here. So 2018 we are somewhere here. So and pathways worked out. Now you just got to accept that this climb is very steep as you go up to the top, right? That's what you would expect. Well, I'm sure we'll get there someday and wave the flag, right? Okay, so now we are ready to do the computational pre-DB protein level bank, pretty much ready. So in fact, we have done it already for PYBAX. It's published in 18, and another strain PVP01 in 2021 in the database journal. Now, but we are trying to work on the human genome now. So that's just Bhagirath server. You just upload your sequence or cut and paste, and you will find that you get a 3D structure depending on the queue. There is a queue always, so within a day. But if you don't get an answer within a day, then you should just email it to me or to the user, to one of these guys. It's help. There is a help site available for this. Get a structure. So I've said a lot about structure, but structure alone may not be sufficient. You have to worry about the dynamics as well. That's where MD simulations come in very handy, right? Structure and dynamics can help function of the proteins, right? That's a conclusion here. Now, briefly, now all of you must be super saturated by now with too much genomics and proteomics. On the drug discovery part, what is it that we have to offer to you in the Sanjeevani part? So the methodology of Sanjeevani is that user, this is a candidate molecule coming from a database or from a user. Remember, million molecules, etc. These can come here, or you can even draw the structure. Then you can put them through drug-like filters. These are so-called Lipinski filters. And then you geometry optimize, or you would assign quantum mechanical charges. So you're preparing the molecule for docking, active site identification and docking, and then minimization complex, MD simulations, and eventually free energy calculations, and finally how good is a molecule for ready for testing in the laboratory, right? So this is the current status of the Sanjeevani. The You can give the DNA as a sequence, as a target, or you can give protein sequence. If you give a sequence of the protein, then it goes to Bagheera to give a structure, right? Or if you give a structure, then it's easier. If you give a structure with active site defined, it's even better. This is what protein ligand complex is doing. That it already tells you this is where you have to bind. Give me a molecule that replaces the ligand. So that will be easy, easy portion, right? So that is the target side. Now on the drug side, well, you can do million compounds, you can do natural products. Now these days we have some Indian medicinal plant library also added to this. You can draw the molecule, then you check for the Lipinski compliance. This is for bioavailability. And then you can do a rapid scan against million compounds in minutes. After that, you optimize, you do the charges and then assign parameters, you do docking and scoring, and eventually MD simulation. Finally, you are ready with molecules for testing, right? And of course, what do you do with the MD simulation? After you do the MD simulations, you extract free energies utilizing some thermodynamic cycle like this, right? And you can also do off-target binding. You can take a good molecule like drug one has to bind to target one. If you get a blue color, it's good binding. If you get a red color here, like target two, target three, it should not bind. So this is a good drug molecule. It's not binding to other targets. Whereas drug 14 is not such a nice molecule. It's like a promiscuous molecule. Target 14 is binding to target 14, drug 14, but it's also binding to target 1. So this can cause toxicity, right? So you can scan off-target binding and toxicity related to off-target binding using computational methods, using in silico methodologies. So that is the idea of this slide, right? Now, what are the success stories? So this is an anti-cancer molecule. So when I say success, I mean it's computationally predicted and it's validated experimentally, right? So this is binding energy calculated and this IC50 is the experimentally measured. So what we are getting is a micromolar compounds. So micromolar is routine. I was mentioning to you in the initial slides. So you can get a nice micromolar compound. It's a very good start for a low micro. Not some drugs, in fact, do bind with micromolar affinities, right? These are dissociation constant. If you get nanomolar, they are very tight binders. You don't have to have a tight binder every time. That's my essence. But it's a good start. Micromolar is a good start. So this is for estrogen receptor. Now, this is for anti-Alzheimer's. Acetylcholinesterase is a target, and you have some molecules here which are binding very tightly. Again, micromolar compounds, and they are validated experimentally. Now, this is anti-malarial N-methyltransferase, Sanjeevani success story 3. Once again, we have prediction followed by experimental validation of micromolar compounds. And in fact, with this, we were able to touch some low nanomolar as well. Right now, antivirals, picorna 3C viruses, we were able to show that this particular molecule binds to 3C. This particular target, in fact, shares a structure with the coronavirus. So we thought that this molecule, some of these isotin molecule that we're developing, could also, bind to the coronavirus proteins. So that was the expectation. Now, collaboration with medical schools. So we have done some antifungals here, which has gone to Sanjeevani. So I was Nature. I was very happy to see Sanjeevani in Nature in 2016. And then there's a lot of drug repurposing work going on in collaboration with the NCI. So that's about the story of the Sanjeevani part. Now, my students said, well, these days you have to have an app, right? I, except that it doesn't work on my phone though. This is only for Android. I have an iPhone, so this works for Androids. So you can download the Sanjeevani app and do the drug discovery while you are traveling in metro or traveling in a train or plane or waiting for the plane to arrive, etc. And what can it do? Well, it can do a lot of these different things. not on your not on your laptop or not on your mobile phone. All these things are happening at the SCF bio computers. So all you are doing is submit a job. Calculations are done on the supercomputer and you get the results. You can fetch them and you can visualize them on your screen. So that is the idea of these Android devices, right? So where are we with regard to drug discovery? A, D, M, E, they're conquered. T is not conquered. Absorption, distribution, metabolism, excretion. Toxicity is a tough part, right? If you can conquer it, then we have done fantastic job of drug discovery. Right, so what I mentioned to you is this Dhanvantari software. Now, this pipeline is published recently, a few months ago in this chemical engineering, which has this chem genome to Bagheera to active site finder to million compounds, RSPD to power dog, atomic level docking and filters, and eventually a potential drug that can be tested. Right. So I'll mention leave this aside right now. There are some molecules which are being waiting for testing. This is FDA approved drugs. So we have some predictions from FDA for these coronavirus proteins, isotyne based molecules, Indian medicinal plant molecules, zinc compound molecules. So hopefully we will be able to do some testing and request uh, people to take it forward in terms of clinical trials and so on. That's where things stand. So that's a, fantastic brain power that I have as well as the computing power, right? Thank you all for your attention. So many people. I summarized you about 25 PhDs work, So that's a, I'm sure you have observed some of it, right? Acknowledgements and thank you very much. Professor Kumar,
Yeah,
take
uh,
over.
thank you so much, sir. Like it was very informative, interesting, and I think it was a more of a revolutionary talk in terms of
Mhm.
like the basic concepts which we have learned over the years. But now, like there is a new paradigm shift. I think it is about to begin because of your hard work, where like biophysical characteristics are going to play a major role. So I think there are a lot of questions which participants have asked, and I will be sharing with you.
Can I start with my question, Dr. Yatindra?
Yes, ma'am. Yeah, yeah, sure, ma'am.
Jayaram, this is Sonu.
Hello, Professor Bhatnagar. How are you?
Uh, First of all, thank you for a very fine insight into how you're using chemical and chemical properties for at the interface of biology, computer science, and mathematics. So it was very interesting. I will put a like big question. In this particular FTP, we have been discussing that how very different types of disciplines go into drug discovery.
Mm.
And when we, like, since the time that I have met you and I've seen you working, you've always had a very big vision about how things should be done. You put up a supercomputer facility, made it available to students all over India to do their work. You made up a pipeline, uh, various things you put together to make a very large initiative. So is it time for us to now move into a drug discovery consortium where all of, like you said, biologists, chemists, computational biologists, and experimentalists can come together in one place?
I agree. I'm 100% with you on that proposal. I think it's about time. We developed these large teams of people together. For example, there are people who are extremely good with the quantum mechanics, right? Even in the computations, apart from what I mentioned, like MD simulations, that most of it is scoring, docking, these are all done using molecular mechanics. There may be some people who are good in quantum. Similarly, there are some people who are extremely good with machine learning. I may suggest a particular docking uh, pose or some such thing. Machine learning people may modify it a little bit, right? So there's another angle from a computational point of view. And now none of this is going to be a success story unless there is an experimentalist who can look at protein ligand binding uh, or cell-based assay, right? So the several ways of experimental characterization validation. If all these people are working not necessarily under one roof under the pandemic situation, but there is a consortium, then promising a molecule is much more convincing or is probably much more feasible. You can definitely come up with 4 or 5 molecules which can be tested, and all this can be done within 6 months. And it's not like for it'll take forever, 15 years, and $2 billion type of thing. That probably is not required. If a consortium is operational with collective brain power, I believe, whatever the disease, with 6 months is about the maximum time that you will require to be able to get. Beyond that, of course, there will be animal models and so on. So there will be a lot of biology that has to be done. But at least coming up with candidate molecules, is feasible, very much feasible. As you rightly pointed out, this consortia approach is the way of the future, right? It has to be. Otherwise, we are working in our own silos, right? So there is a problem, right? Agreed. I hope some of you can trigger this kind of consortium approach.
Sir, there is a question from a participant from DIT.
Yeah,
Whether Sanjeevani is good enough for both small molecules and peptides with molecular weight 200 Dalton for
yeah,
docking.
yeah, yeah. It is. It can handle. It can easily handle. Yeah, for small molecules, it can handle. In fact, now this NSM under the DIT DST initiative, NSM stands for National Supercomputing Mission. What they've asked me to do is to port the Sanjeevani on the national network so that anybody from anywhere in India can access the Sanjeevani with the computational power of the NSM, National Super, which is running into petaflops very soon, right? So people with all you need is some kind of a connectivity that Garuda, that NKN network. If you have that NKN network and if you can hook onto the NSM machines, then you should be able to do a lot of things. Sanjeevani is one of them, right? So that's what we are working on. Hopefully within this 1 year, we should be able to port all the Sanjeevani modules in a user-friendly manner to people so they can utilize it, right? And of course, our lab is always there to help clarify any questions and so on, yeah, to make it easy for users to use it, right? Yeah.
Sir, there is another question which is asking about do you have any server or you can suggest study to do the cytotoxicity studies apart from admit properties?
We have been working on these toxicity filters for a while. Somehow we have never finalized them as yet, but I'm not convinced because they have to work every time. Right now, experiment is the only way. So whenever my experimental collaborators are measuring the protein, whatever I predict, they're buying, they're testing at a protein. So they do the cytotoxicity as well. So is it toxic to the host or not? Or is it toxic only to the target protein kind of thing? So as of now, we are going by the experiment, not by predictions for the toxicity. Although there are some softwares, we have not put up a toxicity software as yet. We are planning to do something, but it's a challenging problem. It's not that easy, right? Toxicity is not that easy, right?
And so majority of the questions are regarding whether these servers are freely available for
Yeah,
users.
they are freely available and people can access them. No payment required, right? Sometimes they don't even have to declare their identity. They can just submit a job and get the result. But if there's a waiting time, then it's better that they give their email ID so that results come to their email ID. So that's their ID. They're free, freely available.
So now there are no more questions, and I'm really happy and really thankful to you. You accepted our invitation for this FDP, and thanks a lot for very
Okay.
revolutionary talk, I must say.
Yeah, thank you, Professor Kumar. Thanks a lot. Very nice. Uh, Thank you, Professor Bhatnagar. Thank you all. Thank you to the audience as well. So it's wonderful. I hope some of you will take up the questions that are left as pending to be answered here, right? So and take the science forward. Good luck. Take care now. Bye.
Thank you, sir.
All right. Sure.
So the next session will start at 1:30.